Okay, so this video is over chapter 12, and we'll be talking about the chromosomal basis of inheritance. So, the chromosome theory of inheritance was created after Gregor Mendel's experiment, but what it proved, or what it says, is the same as what Mendel's laws were. So, they're very, very similar. The chromosomal theory of inheritance is genes have specific locations on chromosomes, which are called loci, and that it is chromosomes that segregate and assort independently. What Mendel said was pretty much the exact same thing as that. Mendel's laws were the law of segregation and the law of independent assortment. The law of segregation states that two alleles for each gene separate during gamete formation. This is explained in the chromosome theory of inheritance because it says chromosomes segregate. The law of independent assortment is alleles of genes on non-homologous chromosomes assort independently during gamete formation. This is also said in the chromosome theory of inheritance. This graphic shows both of Mendel's laws of inheritance in action. So, sex-linked genes, sex-linked inheritance is important in this chapter. A sex-linked gene is one located on a sex chromosome, either the X chromosome from your mother or the Y chromosome from your father. Thomas Hunt Morgan discovered the existence of sex-linked genes, and he did that through the Drosophila fly. The two types of sex-linked genes are X, female, and Y, male. The majority of genes that are carried on the X chromosome are not related to sex. If these genes are a little bit off, this can cause Duchenne muscular dystrophy, which is the weakening of muscles and loss of coordination. Hemophilia is the inability to have normal blood clotting. That's also on the X chromosome. In females that have two X chromosomes, one becomes inactive during embryonic development. This is called X inactivation. These inactivated chromosomes are made this way through methylation and condense into bar bodies that lie along the nuclear envelope. Sex-linked genes calculations on this last slide are similar to the Punnett square, but they're obviously a little bit different, and you can see that there. The alteration of chromosomes cause genetic disorders in a human being, in an organism. Non-disjunction occurs when the members of a pair of homologous chromosomes don't separate properly during meiosis II. Non-disjunction is shown right there. If the faulty gamete with no copies of the chromosome engages in fertilization, the offspring with an incorrect number of chromosomes is called an aneuploidy. The offspring with an incorrect number of chromosomes is called aneuploidy. Eggs with 3 copies of a chromosome are trisomic, which is shown here, and eggs with 1 chromosome are monosomic, shown there. Polyploidy is the condition of having more than 2 complete sets of chromosomes, common in plant species, and that's part of non-disjunction. Errors in meiosis can occur in many different ways. The first one is deletion, a part is removed. Duplication, where the same thing is coded for twice. Inversion, where 2 things are flipped. And translocation, which is where 2 parts of genes are kind of swapped. Human disorders that are caused by non-disjunction are Down syndrome, This little baby over here has Down syndrome. Klinefelter syndrome, which is where a man has female qualities, and Turner syndrome. Genomic imprinting and the inheritance of mitochondrial DNA is important. Genomic imprinting is traits differ depending on which parent passed along the allele for that trait. That is shown in the picture. Mitochondrial DNA only comes from your mother, and they got it from their mother, and they got it from their mother. Mitochondrial DNA only passes through the mothers. Good luck on the AP exam.